Okay, hi everyone. Let's start. So we're happy to have today Dr. Omer Revah from the School of Veterinary Medicine. Omer started his career in the School of Veterinary Medicine, doing a degree both in VDMD, how it's called? DVM and PhD, working on cortical computation. After his PhD, he moved to Stanford to work with Giorgio Pasca. And I think part of the work that he's going to talk today is related to that. So Omer is doing something, to me it seems like close to science fiction, where they take stem cells from animals, they grow little organoids of brain, and then they implant them in the brain of other living, behaving animals, and they measure that. So for me it sounds like science fiction, but hope today you will turn it into science. So thank you for being with us.
Thank you, Jonathan. Thank you for inviting me. Can you hear me clearly in the back rows? Yeah? Okay, cool. Yeah. It's a recording microphone. There's no...
Speak a little bit.
Okay, let me know if I'm becoming, if I start mumbling or something, so I'll turn my voice up again. Yeah, feel free. I started my scientific career in the lab of Mike Gutnick. We're the stepsons of the Neuroscience Society of the Hebrew University back in the Rehovot campus. And as I was about to finish my PhD, I was looking for what I want to do in my when I be a grown-up in my long-term career. And I really looked into and wanted to start studying neurodevelopmental disorders such as autism spectrum disorder. But because I already knew a little bit too much about the brain, I also felt deterred, and that the task is kind of huge, and understanding the brain is really hard. And I don't know if you know, but if a child is diagnosed today with a neurogenetic disorder, that in 5 or 10 years will be quite harsh for him and the family, there's basically 0 treatments to offer. And in this paper here, it's working good. They even try to quantify this. If you're a startup company, basically the last thing you want to do and explore are drugs for neuropsychiatric disorders, and especially developmental disorders. So it's negatively correlated with the success of the company. But at the same time, more or less, I heard about a new technology that really fascinated me. And I heard about it in a conference in Israel. Basically, the idea is that you can take fibroblasts. Maybe some of you already heard of this. You start with a skin harvesting of fibroblasts, and then you do a step called reprogramming, where you take the fibroblasts sort of back in time, you make them into stem cells, and then you can you differentiate them into neural structures, also called organoids. And importantly, you can take these fibroblasts from any individual, including people with specific neurogenetic conditions, with the hope that this will help us learn something about their disorder. So a little bit more about how this is done. In this case, this is a cortical organoid, which is what I've spent most of the time focused on. So again, you start with stem cells that were skin cells in the past, and you aggregate them in this thing called an aggregate well, and you make these little balls of stem cells, and then you start a differentiation protocol where you try to push them into a specific lineage. And the brain has multiple lineages, so cortex is one of them. Cortex requires the least effort. It's actually the most well-studied one and most well-understood. And this process called dual SMAD inhibition pushes them directly to cortex. So the lineage is actually working quite phenomenally well, where you get very specific patterning of the cells. If you look at the plate, you can see here at day 0, these are stem cells in this aggregate plate. And after 6 days, you detach them from the plate, and they start to be, they free float in a 10 centimeter petri dish. and you keep differentiating them for a few weeks. And after a few weeks, is it working? Like this. You get this human cortical organoid structure, which is comprised of different cells that are classical for the developing cortex in the first few months. So that would be radial glia cells, which give birth to the neurons and astrocytes. And you can even get some deep and superficial layer neurons. Yes, and you can even do some electrophysiological recording from them, from the neurons. You can see that the fire action potentials, after you block sodium channels with TTX, you can block the action potentials. So in the first part of the talk, I'll talk about how we use this system to do in vitro phenotyping of a human disorder called 22q11 deletion syndrome. And in the second part of this, I'll try to show you some of the recent work that was just published, where we take this system a step further and try to build a model that is a cortical organoid model, but this time we do it in vivo. All right. So a little bit about 22q. 22q is a syndrome, which means that it affects several aspects of physiology, not just the brain. But it's interesting in relation to the brain because it seems to be increasing increasing the likelihood to be diagnosed with many of these conditions, including ADHD, ASD, and it's mostly known for the fact that it increases your likelihood to develop schizophrenia at the later point in life. So we wanted to use this new system, and we, in the first step, we generated the largest cohort to date of stem cells from patients with 22q11 deletion syndrome. So we had 15 controls and 15 patients, and we differentiated them into organoids. We programmed, differentiated them into organoids. In the first step, we did some kind of a sanity check, where we look at this deletion. It's a microdeletion that involves between 40 and 60 genes. And we found that indeed those genes, because it's haploinsufficiency, meaning one of the chromosomes is affected, we saw a reduction of about 50% in gene expression. Next, we did a gene set enrichment analysis, where you kind of take the cells, you look at their RNA data, and you find what kind of terms. There's like a black box algorithm that shoots these terms. And we found that many of the terms that are associated with the disease seem to be related to calcium channel activity. And then the next step was to try to look at calcium in the cells. So we did a simple experiment where you take the organoid neurons, you fill them with a calcium indicator dye, and you depolarize them with KCl. And what we found very robustly across different lines of patients is that they have a reduced response to KCl. And this was very interesting, and we're looking at this for a while, but we wanted to understand why. So the first thing, the most obvious thing was maybe they're just lacking calcium channels, voltage-gated calcium channels. So we recorded from the cells, but we couldn't find any evidence for that. Their calcium channel densities were normal. We then looked at, we patch clamp recorded many cells from many lines, and we couldn't find anything that popped out. Actually, most of the features were completely normal. There was one thing that we found that was different, and that was the resting neuron potential of the cells. So organoid neurons seem to be sitting around minus 50 millivolts, so they're quite depolarized. But in the 22q cells, we found that the cells are depolarized by about 10 millivolts. And again, we did this across different lines. So then the idea was that maybe if they're sitting depolarized for a long time, maybe they have, this causes their calcium channels to be inactivated, which is a state in which the channel is in a state where if it would get an activation trigger, it can't open. And although the inactivation properties seem completely normal, if you plot the resting neuron potential of the 22q neurons, you can just explain this reduction in calcium flux. And here we did an experiment where we more closely studied and we filled individual neurons and we have perfect control of the resting memory potential. And we found that here you can see how the neuron responds with a calcium increase following a depolarization. And we found that if we give a small step of depolarization before the stronger depolarization, you get a decrease in the calcium flux. So again, because this system is quite nice, within a few weeks we said, okay, let's try to find which of those genes in the region can explain the phenomena. Maybe it's one gene, maybe it's 40 genes. And we had a reason to suspect that GGCR8, which is a microRNA processing protein, is implicated because there were evidence about this. So quickly we did we generated a clone where DGCR8 is deleted. One copy of DGCR8 is deleted, and we repeated this calcium imaging experiment, and we found it did a reduction in the calcium flux. Not only, we even repeated the resting memory potential experiment, and we found that the cells are depolarized. And as the last experiment, what we did, we tried to rescue the condition by overexpressing DGCR8 in the patient cells. And we're able to repolarize them sort of back to the normal, quote-unquote, resting memory potential. So this is just to illustrate some of the things that the system can do, and I hope I convinced you that the system can be quite useful. But in the next part of the talk, I'll try to convince you that I'm wrong.
I can ask a question. So this is really cool. What do you think but the connection between DGCR8 is? It's not obvious. It's not like a calcium channel that you found.
Right.
So
Right.
What is the hypothesis?
So there is a hypothesis. It wasn't in the final it didn't appear in the final manuscript. We did RNA sequencing. We compared RNA sequencing of the two conditions, and we found that there is convergence on a few genes, and the most interesting one of them all was ATP1A2, which seems to be expressing in the young neurons and is reduced in the DGCR8 network. So the idea is that without this, it's a sodium-potassium pump. The idea is that without the sodium-potassium pump, there could be change in resting memory potential, but we didn't follow up with this.
So it's changing a microRNA. The microRNA is regulating
right.
the pump.
DGCR8 is changing all microRNAs, right? But somehow the pump seems to be conserved across the two conditions.
So DGCR8 is sitting in 22q11.
Sorry?
Is it one of the genes that is sitting there in 22q?
Oh yeah, so DGCR8 sits in the condition. Yeah, maybe let me show that. Because I think it's interesting. Probably it's one of the things that I'm supposed to hide, but I actually think it's interesting. DGCR8 is one of the genes in the region, and it's actually this gene here. So if you see a 50% reduction in all the genes, DGCR8 seems to be the most regulated one and most compensated one, which maybe points to some of its importance. We couldn't explain this, but it's still statistically significant reduced, but yeah.
So you think the other genes are less relevant?
It seemed, at least for the phenotypes that we identified, there were dramatic differences in the sort of transcriptome of the two conditions. So it's not mimicking the syndrome in full, but it seems to be important for this condition, for these phenotypes. Good. So what's the problem with cortical organoids in vitro? So although they are really, really powerful, that's true, and they're very flexible and scalable, one thing that is true for all organoid models is that they invariably fail to mature in vitro. So they're really, really good in mimicking the first few months of development, 2, 3, 4 months, but then they don't seem to follow with the normal developmental trajectory in terms of maturation, neuronal maturation, specification of cell types, and other things. Also, cortex in a dish just sits isolated like this, and it doesn't receive any input or sends meaningful output to the brain. So our approach to sort of overcome this was to take the organoids and transplant them into the brain of newborn rats. So the way we start this process is same thing. We start with stem cells, we do a cortical differentiation, we come up with human cortical organoids. But then at about 30 or 45 days of differentiation, we take them and we inject them into the somatosensory cortex of newborn rats. And the rats are also immunodeficient so that they will not reject the graft. And this is how it looks in MRI. We think because this is embryonic tissue, the water content is higher, so that they kind of pop up in MRI because they're brighter, into T2-weighted MRI. And what was really nice is we could do this across many, many lines and conditions with very high success rate. So in a way, although it sounds like you add a lot of complexity, once you have everything figured out, it can save a lot of work, because media changes for in vitro cultures are really, really demanding. So once you put it in the rat, when the rat is a few days old, it will basically culture your cells for half a year. And one of the first things we noticed is that, this is THCS, so transplanted human cortical organoid. Ignore this. So one of the things we notice is that dramatically grows after transplantation. So if we start with an organoid that maybe will be a little bit smaller than this, after 5 months after transplantation, we get these huge tissues. And it was even a little bit concerning, where it's like, oh, well, what are we growing in here? Another thing that happens really, really rapidly after transplantation is vascularization. So I'm sorry for the bad aliasing here, but I try to pop up the color. So this is staining for rat endothelial marker. So it's rat specific, and what you see is that the rat immediately starts to vascularize the graft. And also there's microglia, which are marked by this EBA1 marker. They're also coming from the graft, from the rat. They're of rat origin, and within a few months they will populate the graft. So yeah, just to make it maybe 100% clear, is that microglia and endothelial cells are not born in the cortex. When the cortex is generated, all of the cell types it comprises of are progenitor cells, all sorts of them, neurons, astrocytes, and oligodendrocytes. So these cells are invading and populating the graft. And this shows a slice of the graft. This is the graft here, and what you see is that microglia are populating the graft, and in blue are astrocytes, which we'll need to see, but they're basically of human origin. And this is another immunostaining where you stain for neurons, and you can see that most of this transplant is comprised of neurons. If we look a little bit further, we find all kinds of cells that we expect to find in the developing cortex: progenitors, neurons, astrocytes, and microglia that are not human, or not of human origin. But we wanted to look at this with single cell resolution. So we did a single nuclei RNA sequencing experiment, where if I'll just try to put everyone on the same page, you try to look at all the nuclei in your tissue, you harvest the tissue, you put it in a small Eppendorf, you mix everything up, and then you get a soup of nuclei, and you take this nuclei and you send them through sequencing, such that each cell, now you get the cell, and you get how much expression it has in about, let's say, 5,000 genes.
I may have missed it, but is there a difference between the age of the neurons in the graft and the age of the other cortical neuron in the rat?
So the rat is, let's say, 4 or 5 days when we transplant. The graft is 45 days from differentiation. So ages are a little bit different here, because let's say for the rats it's 26 days from start of gestation, and for the human cells it's from differentiation. But differentiation sort of skips about a few weeks in gestation, because when you start gestation, corticogenesis doesn't start immediately. But the idea is that they kind of develop at different rates entirely. Human cells develop extremely slow. Rat cells are developing much faster.
Did you tell us if these rats had some human capability?
Yes, I didn't. They don't. They don't. In the first few months, I was walking into the animal room, and I was just hoping that none of them will speak to me and say, "Why are you doing this?" But it didn't happen. They were looking at me, but no language. So with this experiment, you can then take your data and you do some kind of high-dimensional analysis, and you get this UMA projection where the different cell types are clustered into, and here they're annotated by names at different colors. And what we found quickly is that you have a cluster of astrocytes, a cluster of oligodendrocytes, and the cloud of glutamatergic excitatory neurons, which are the three lineages that are born in the cortex. And some of the oligodendrocytes were even myelinating, which was really, really, really interesting. We then took and did a comparison about the same time. We took organoids that were kept completely in vitro during this time, and they had roughly the same cell populations. They had astrocytes, neurons, and progenitors, but they didn't have any oligodendrocytes, and they had this cluster that sort of isn't supposed to be there, supposed to be there, which are interneurons. So interneurons in development are born in the ganglionic eminence somewhere else, and during development they migrate and populate the cortex. So this was really, really interesting, and mostly we looked at this shape of glutamatergic neuron clusters, and we saw that it seems to be way more complex than the glutamatergic neuron clusters that we find in vitro, although we could define them into upper layer and deep layer neurons broadly, but this seems to be more refined. So we try to look at this more closely, and we took this glutamatergic neuron cluster and we try to map it into a single cell atlas of human cortex. And basically this is what we found, that the different clusters seem to be mapping really well into the clusters into clusters of human cortex, which are corresponding to some of the layer markers that we know, like layer 2-3, layer 5 cells, deep layer 6 cells, and so on. When we try to repeat this with the in vitro organoids, we couldn't find such refinement. There seemed to be maybe some signal in there, but not much. Then we also wanted, right, and this explains this a little bit, shows this a little bit in more detail. So these are the transplant clusters, and these are the LN brain atlas clusters, and you can see that for some of them there seems to be almost one-to-one matching, and this is another atlas that also shows high matching, but the in vitro organoids, a few of them match, maybe one astrocyte marker, meningeal cell marker, but much less. I think it's clear. We then looked at maturation. We did a bulk RNA comparison, and we found that many of the terms, similar to this gen seed enrichment analysis, many of the terms that we find that are upregulated in vivo seems to be related to neuronal maturation. So those are synapse, dendrite, axon, and other genes. And usually people do a lot of cherry picking for this marker, for these terms, but we actually had like a perfect list. Everything was neuron. So we said, let's try to do some recording and morphology comparison between the two conditions. And we were quite shocked when we found that in vitro neurons, maybe they are kind of pyramidal in shape, but when we look at them in vivo, we find these huge morphologically complex cells with dendrites and dendritic spines and so on, everything you can expect from a pyramidal cell. This is quantification of morphology. And like you can see that total length is maybe 20 times larger or 20 times longer in the in vivo condition. And also dendritic spine seems to be actually growing, and this we really had to fight to find one example in vitro that shows nice dendritic spines. And this is also quantified here. And also in terms of this resting mirroring potential, I think you saw before that normally in vitro they sit at minus 50 millivolt, but in vivo they sit at much more polarized potentials. Yeah.
Maybe obvious, but how do you know if you're going from human or rat?
Yeah. Good. So yeah, I should have said something. So first, for the single nuclear RNA sequencing, there's also some red cells in there. So you do kind of transcript mapping, and you sort of throw all the red cells. It's a very clear cut. For this, the transplant is very, very large, and what you see is that there's no rat neurons migrating into it. Like never. We did a quantification for this. I can show you in a second. Then we also did some optotagging experiments for the in vivo recordings, but it's clear it's all human. There's maybe 1 or 2 rat cells that are sitting at the border. By immunostaining, you can see it, so it's clear.
So you think that all the synapses that are made here are made from human cells to human cells?
So
there's no interaction between human cells and
I will touch on that on the second part of this study. There is interactions, but to your question, most of the connections are human-human and not rat-human.
And this is in the barrel cortex?
We transplant in the barrel cortex, right, in somatosensory cortex.
Can you get to respond to transcriptional?
Yes, but this is coming up. So, and if you do a patch clamp recording, you push some current into the cells. So you find that the in vitro cells, they do fire a little bit. They can accept a few picoamps of current, but then they kind of collapse and they fall into sustained inactivation, while the transplanted cells just respond with beautiful action potentials, which even kind of, if someone look a little bit like the ones that are supposed to be in a pyramidal cell. And finally, we wanted to see, we got a comment from one of the reviewers, but where do we stand in the maturation state of the cells? How mature are they? So we transplant them for maybe 6.5 months and have been outside for another 1.5. So they're about 8 months in from differentiation, and we found that indeed they fall exactly on the early postnatal or just a few days before birth or after birth of a human. And this is in terms of transcription. Then we did another experiment. We also found this with the morphology analysis, where you find this is a human juvenile layer 2-3 pyramidal neuron that we filled, and comparing to the transplant, it's still larger. This is a Scholl analysis where this is quantified as a complexity of the dendrites easily, and you can see that the transplant sits lower than the 2 human samples that we looked at. But interestingly, the electrophysiological properties were not behind. They were already there. So the cells were completely polarized, they were firing normally, and the spikes were as sharp. So just quantify here with spike half-width. Another finding from transcription was an upregulation of activity-dependent genes. And those genes are important for maturation, important for plasticity, and many things. So in the transplant, we found a strong increase in activity-dependent genes, both early and late. And we wanted to use this to do some disease modeling. So there was one disease that we could think of that had something to do with activity-dependent gene expression, and that's Timothy syndrome. So Timothy syndrome is another disease associated with autism spectrum disorder and intellectual disability, and it's caused by a point mutation in CACNA1C. And in contrast to the 22q, in this disease you get an inverted picture. So because the mutation sort of stabilizes the inactivation properties of the channel, such that it doesn't inactivate. So when you try to depolarize it with KCl, you get sustained flux of calcium. So we repeated the experiment from before, but this time we also transplanted organoids, so again, this is HCO, from people with 3 individuals with Timothy syndrome, and we repeated some of the morphology analysis, and we found that there was a huge reduction in the size of the cells, and as quantified also here in the complexity of the dendrites, but there was an increase in the number of spine and spine density in the disease. We could also take it a step further and also look at some maturation-related electrophysiological properties. So altogether, this, I think we really showed well that this system can be used for disease phenotyping in a human context. And much better so maybe that we could do in vitro. And this is just examples of some of the morphological reconstructions we did from people from cells from Timothy syndrome. And I don't know, I think this, I hope no one ever is diagnosed with this because it seems like super harsh. Yeah. And importantly, when we try to do this shoal analysis that compares control and Timothy syndrome, when we did this in vitro, we couldn't find any differences because the cells are not morphologically complex. So the question the elephant in the room, because this is ELSC, everyone wants to know if it communicates with the rat. So yeah, basically yes. But we try to look at this also very systematically. We first did rabies tracing. We wanted to see which neurons from the rats send axonal projections into the human graft. So you infect the organoid with the G-deleted rabies, and you supply the G protein. And after a few weeks after transplantation, we found which areas, in the rat brain, seem to be projecting into the rat. And because we transplant here in S1, it seems to be quite specific. So in S1, most of the input comes from the thalamus and from the somatosensory cortex of the same side, and maybe a little bit from the other sides, from the other side. So this was, again, a good sanity check to see that we're connecting to the right places in the rat brain. And then we did something called slice recordings, where you take the rat brain with the graft, you make coronal slices. In this case, it's thalamocortical slice, and we can stimulate axons coming from the somatosensory cortex of the rat, and axons coming from the internal capsule of the thalamus, and we saw synaptic responses in the human neurons. We then also did we infected the rat thalamus with carmine, which is some kind of an opsin, and we found that following light stimulation, there's a small response in human cells. Then we wanted to look at this in vivo and see what kind of network behavior or network activity we see in those grafts. And this is a 2-photon movie of GCAMP activity in the graft, and you can see that they're very synchronized and seems to be going on and off all the time. These cool things here in the back that are moving slowly. I don't know if Inbal is in the audience, but these are astrocyte signals. So this is a GCAMP was on an EF1 alpha, so it wasn't selective for neurons in this case. This is spontaneous. Then we wanted to see if it does receive connection from the surrounding, if it's physiologically active by sensory stimulation. And we repeated the experiment, but we gave a small deflection to the whisker with a piezoelectric, and we found that indeed the cells respond within a relatively short time scale, but when we do randomized time steps, we can't observe this. We then repeated this with the multi-electrode array recordings, and we saw exactly the same phenomena. It was quantified across multiple animals. And this is for this, right? Recorded cells are of human origin. This is true also both for the patch clamp recording. So you can see this is a human nuclear antigen staining, and you can see that the borders are quite clear. I don't know if you can see that or not. But when we quantify the number of neurons that are not HNA positive, they're basically zero. All of them, within the graph, they're all human. All of the neurons are human. And we also did optotagging experiments where the cells express channel rhodopsin, and then you can shine a light and see whether your cells respond with a short time latency.
Yeah. So those responses you showed for this integration, so can you say a little bit more about the features of the response? I mean, is there a map or
So they respond with the same behavior as you can see in the video. It seems to be an all or none synchronized response. But with the whisker activation, you can sort of move it in time such that it comes right after the stimulation. Sometimes maybe it fails if it comes within a wave.
They respond to all whiskers, only to one of them?
We put all the whiskers into the thing, and so we didn't try that. But my guess looking at the map is that they will do, if this specific barrel is not innervating somehow, or barreloid, right? So maybe it won't. That's why we try to put all of them. Yeah.
So actually a follow-up question on that. So you said that you couldn't identify interneurons in this transplant, right?
Right.
Wouldn't that somehow affect the tuning properties of these neurons?
Right. I hope you'll be the reviewer of my ISF. So yeah, they're missing. So interneurons are born somewhere else, so we have to actively place them in the cortex, so allow them to migrate in. Right. So this could explain the behavior, but I don't know.
Like the behavior?
The network behavior, right. So at the last step, we wanted to also look at the flip side of this, because those grafts also project and send axons throughout the rat brain. So we try to trace that by just using a EYFP on a channel rhodopsin, and you can see that this is the graft here in this slide, and you can see axonal projections in multiple brain regions in the rat. We quantified that. There seems to be some consistency, but it's not 100%. It seems to be strongly projecting to the rat brain. And next, we even try to see if those connections are functional and monosynaptic. So we recorded again in slices, and this is a recording from a human channel rhodopsin expressing cells, and this is kind of like the channel rhodopsin current respond to blue light, and here you can see that it's spiking. And when we patch from rat cells and we give this strain of stimuli, you get synaptic activation, which if we slightly depolarize the cell can even, in this recording, cause spikes. So we think that the connections are there, but they're also functional. But the major goal of this system is, well, we want to study human brain development, but we also want to use it to understand disease better. And most of the diseases that we try to understand are basically emerging on some kind of a circuit level and some kind of a behavior level. So we wanted to have a readout that is related to the activity of the graft, but we can read it from the behavior of the animal. So the idea we had was to try to train the animals to know that the human cells are active. So in this case, we again transplant an organoid that expresses channel rhodopsin, and then we start delivering blue light to the rat, and following a few seconds we give a reward, a water reward to the animal that is deprived. And when we give red light, we don't give it a reward. And this is kind of a training and testing at the same time. And what you find is that on the first day, the rat doesn't have any preference for blue or red light. But within 15 days, once you turn the blue light on, the rat starts seeking for the reward. And we measure this by something called a licometer, where you measure the number of times it protrudes its tongue and try to look for the reward.
How does it measure if you just inject the virus directly to the rat?
Very similar, very similar. Yeah, this is quantification of this. Then we did another experiment with them, which I'm not showing, where you see that activation of channel rhodopsin activates by using CFOS staining, activates many places subcortically in the rat, maybe the VTA. We didn't have any idea for what's exactly the mechanism for this learning, but it seems to be, if you can get it from the cortex, maybe this explains it, because we show that it directly activates the cortex. So just to quickly wrap up on this second part of the talk is the unique properties of transplanted human cortical organoids is that they're efficient, scalable. We have a cellular composition that is very closely related to human very early postnatal times. upregulation of activity-dependent genes, and so on. And finally, of course, bidirectional connectivity with the red brain. So I don't know if I'll have, I think I still have time, so I'll just give some kind of future directions for where I want to take this system. And as you mentioned before, this activity, here it's recorded with multi-electrode arrays. You can see that the activity is super synchronized. And one option is that who are we missing here are interneurons, which should be in humans even more. They're about 35% of the neuronal population in the cortex. So we're completely missing them. And the idea is, right, and this I explained, they're born in those ganglionic eminence region, and they migrate long distances to populate the cortex over months of development. So the idea is to repeat this experiment, but this time you make a cortical organoid and also make a ganglionic eminence organoid, which has been already done. And what you can do in vitro is take them, put them together in a small Eppendorf, and the two brain regions we kind of fuse, and those interneurons will start migrating towards the cortex even in vitro. But in vitro, this process is not super reliable. We see that maybe 10 or 15 cells move, and then you have cell death, all the problems that you have in vitro cultures. I think that if we do this in vivo, we'll get much better results. It could be more complicated because ganglionic eminence, as it's shown in this picture, is not a single entity. There's a caudal ganglionic eminence and a medial ganglionic eminence. They might be a reason to differentially parent them to 1 and 2, and then maybe you need to put 3 together. But I think that this is the direction. And finally, one thing that this can be used for, and we're basically already doing this, at least in the Pascal lab at Stanford, is we have a chance to maybe do something about finding drugs for patients. So this is unique time maybe in history because we were not able to do it before. But now for the first time we have access to human genetics. We have access now also to the target tissues we're trying to target, like cortex or maybe other brain regions. And we're able to phenotype them with high fidelity. And at the same time, there's been a huge revolution in gene therapy approaches. And the idea is that we can do in this transplant model drug testing with much greater physiological relevance. And we don't know, they could be coming from the hormonal system, and there's also the connectivity going on. So there's maybe too many parameters for me to try and break it down. But one simple experiment that one can do, at least to rule in or rule out the connectivity thing, is to try to reduce activity in the human cells by expressing, let's say, KIR 2.1 in the human cells and sort of clamp them to hyperpolarization, won't let them participate in activity, and see if that affects maturation. Yeah.
Have you tried to compare the grafting into older mice brains? Do you get different?
So these are rats. Yeah, we didn't try. This was done in old mice. Seems to be working also in old mice. Our hope was that here we get sort of more flexibility in the nervous system to accept the graft, also immune-wise, but also connectivity and circuit-wise. But we didn't directly compare.
Fascinating. So when you remove the graft, so two questions. One, what happens to the graft and what happens to the rat? So
When you remove the graft,
yeah, if you now surgically remove it after 8 months or so, right?
so we sacrifice the rat and we take the graft.
Can you surgically remove?
Yes, you could. I had an idea to try to do some kind of punch biopsies from the graft and try to track it over time.
Okay, so
We
you
could try to do that.
sacrifice the rat. So this is the rat. But in terms of the neurons, so do you still have dividing neurons? Can you culture them after the?
So most of the progenitors we see, at least at this late time point of 8 months, seems to be all progenitors giving rise to oligodendrocytes and astrocytes, and no more neuronal progenitors.
So the neurons are all post-mitotic.
All the neurons are post-mitotic.
And do you have GABAergic neurons?
So there's no GABAergic neurons, right? This was the single cell graph just shows that. I mean, there's a follow-up study that we did where we looked at. This was only 3 samples. We then repeated this with 18 samples, and we found 1 sample where there are a few interneurons. But the general answer is there's no interneurons.
So you think it's the environment?
So
You
we-
managed to
we-
transplant in the striatum?
No, because they're already following differentiation. So if you look at the cortical organoids very early in differentiation, it doesn't have interneurons either. But it seems to be maybe the stress conditions in vitro after long periods, there's a theory that there's ventralization of the progenitors to give rise to interneurons. But there's another study that found that after transplantation, they find fewer interneurons from a human fetal tissue. So when they culture in vitro for long terms, it seems to be nice for a nice environment for the interneurons.
I was surprised. You have, I guess, you have several million of cells in this mature graph.
Yes.
And these cells, this circuit does not have the inhibitory balance, so they oscillate all the time as you show them.
Yeah.
But you said that the behavior, and there are connections between this region to the rest of the brain.
Right.
I would expect that millions of cells oscillating will cause some kind of, I don't know, tremor.
So you go back to your
epileptic seizures or
exactly
something like that. Where is the output of this? Where is the effect of the output of this oscillating big system connected to the rest of the brain of the rat?
Cool. Yeah, cool question. So this was a 3 out of 3 reviewers said, well, do the rats seem to be having seizures because it's oscillating? And you show that they're connected. But as a matter of fact, they're not seizing. We did a thorough analysis, and we also looked at maybe hundreds of individuals. I never saw any seizures. And this goes back to your first question. The connections between rat and human are there, but compared to what's going between rat and human, they're much weaker.
So a follow-up question to this.
Yeah.
Did you try to manipulate the connectivity between the rat and human cells by coordinated stimulations, for example?
By what?
Coordinated stimulations.
Coordinated stimulation to see if they sync up. So when you throw your MEA electrode into the cortex of the rat, you find completely desynchronized activity, which is like what we see in a rat cortex. You put it into the human thing, you start sync. So we didn't do paired recordings, which maybe we would find a signature, but it doesn't seem that everything is propagated in the rat. The rat, we also did a lot of behavioral testing, just general, to see if they have deficits or maybe they start speaking, but we couldn't find anything obvious.
But if you actively,
If you can actively pair them.
like, it's like conditioning, neural conditioning on the cells of the cortex to respond to the activity in the input.
So maybe we're doing this with the reward task already, but it remains to be seen. We didn't try it directly.
Yeah. But it's likely that there is big connection because they are always firing together.
Because the human cell seems to be always firing together. We don't really find a rat.
For the weak connectivity.
I see, because they're weakly connected, they're synchronizing?
No, because always synchronized, we don't develop a strong connection.
So no, the weak connections are only between human and rat. Within the human, if you stimulate there, you get a huge response, and that's why they're all synchronizing. So they're internally connected really strongly, but they seem to be having a preference for the age or for the species.
So many of them are all excitatory, so then they have to be really strong to get a very strong
Right.
synchronized connection. Very weak
So,
connected.
I don't know if I didn't quantify single axon to single dendritic spine connection and how powerful it is. Seems to be relatively normal, but maybe it's weaker than it can be. Yeah.
You showed only the, first of all, it's amazing. And
Thanks.
you showed only the case when you compare this human transparent, like human neurons to like human cultures or human postmortems. Did you compare them to mice? Like how far away are those neurons from the regular mice?
Right. That was another reviewer comment that we ignored. But it's a question. I mean, from it seems to be also lagging behind the rat. It's still not quite there in development. It's just newborn, late postnatal, late prenatal.
Can you do the transplant in the embryo? Then you get
transplantation, genetic transplantation?
We tried to do some in utero experiments, and it was really, really hard. So we didn't try that systematically. But when we tried, usually we were not. And then also we place it within the cortex. If you do the transplantation in utero, it will be maybe if you do it targeted with the 2 photon, because the entire cortical plate in utero is maybe 100 or 200 microns, so it will be extremely hard to place it there. But even then, the neurons, when you transplant at day 40 of differentiation, it's a sack of progenitors and really, really newborn neurons, while interneurons are already fully migrating in the rat. So they're following 2 different time complexes.
So if you implant organoids into the brain, they're kind of taking up space, right? And you said they're growing over time. So are they kind of moving mass from the original rat brain? Is there like a structural difference that shows up?
It's a good question. I think that they create some destruction of the tissue and some displacement of the tissue. And which one more, I'm not sure.
Yeah, so you said that when you would walk in the lab in the morning in the beginning, you were worried that the rat would start talking to you.
Right.
You said that you sacrificed them at 8 months. So humans also don't start talking to you for like 12 months. So if you would leave it on there longer,
So yeah, cool question.
But also it relates also to the connectivity that we asked you about. That if you just keep them living 2 years.
2 years. These rats can live more than 2 years even. But so one question that I didn't get that sort of answers this is what kind of structure you have in those grafts. So cortex has a very specific radialized laminar structure. Here we don't seem to have that in full. We do have some evidence of laminarization and some evidence of radialization in some areas where it's touching the rat. So you can see where it's touching the rat, it seems to be getting enough signals to radialize and organize in space, while other more faraway regions in the graft are all over the place. But in terms of the number of cells that we're transplanting and the fact that the network activities seems to be kind of messed up, I don't think that's a concern that they'll actually start talking. Also, these cells are quite similar to the cells of rat. It's not like they have that talking gene.
Before transplant, there was no organization.
So there is some organization. I showed this here. There seems to be some organization from right here. You can see division where the neurons are born, and then another layer is born. So this is on a small micro scale. So this would be maybe like 50 microns across or 100 microns across. So you can see some separation between deep and superficial layers. It's all radial. But if you look at the organoid as a whole, this is called a rosette, a neuronal rosette, which it doesn't occur in vivo, but it's close to the ventricular zone we see during development. And the rosettes in the organoids, if you look at the organoids as a whole, it has many rosettes. So it has all these like originating units, and they start giving birth to neurons across them in 3D, and then you get an entire mess. Some people are trying to culture single rosette organoids, where you get a little bit more organization, but yeah.
But you said that there's a very structured connectivity with the rat brain, right? So just
Did I say that? What?
The signature for organization would come from this connectivity. It doesn't have to be layers, but
So I think that many of the connectivity principles are likely to be genetic dependent. So maybe we have specific connectivity between layer 4 and layer 2-3 cells, right? But this could be, it doesn't mean that in space they have to be matching and being sitting just above them. But we didn't look at that. So it could be also that it's messed up. I don't know. Yeah.
So I was wondering, maybe Elon already asked this. I was curious, why did you go for the lick task instead of a whisker conditioning task?
Right.
I mean, that would be the most
okay, so
experiment to do, right?
Okay, so we thought that the reward task is the most straightforward, and this is Felicity from the DICE-ROT lab who collaborated with me on this. I assume she is a genius because when I met with her, I said we really want to take it a step further and see whether it can manipulate behavior in the rat. And then, so she said, so does it project out? I said, yeah, it's projecting, but all over the place. It goes to the striatum, goes to the thalamus, goes to other subcortical regions. And she said, maybe they can learn a reward because rewards are overrepresented in the brain, which for me as an electrophysiologist, I said, oh, really, I didn't know that. So maybe it doesn't really matter where it gets the activity. The rat can sort of self-reflect and say, oh, I'm going to get a reward because this kind of activity appeared in the network. So that was the idea, and from what she said, and I take her as the expert, she said this is the most straightforward approach we can take. But maybe I'm not an expert in this, so maybe I'm wrong.
And the reason I'm asking is because you are sort of blasting it with a lot of blue light, right?
Right.
So the neurons are firing like crazy, so some of the projections maybe to the VTA are getting activated. But
yeah, we did this with just with fluorescent reporter experiments like with YFP, and we couldn't find the evidence for learning. So
right. So
yeah.
what I'm curious is because here now these cells are extremely overactivated, out of synchrony, or rather firing completely in synchrony,
right?
Very high firing rate. But if you want to study more cortical-like functions, then you probably have to see whether the normal tuning curve-based firing that you see when your whiskers are tickled, whether that can be somehow
right.
like integrated with the overall brain function or not, right?
Agree. I agree. This would be really nice. We could sort of close the loop here and see whether, like, sort of physiological activation of the cells also can be associated with learning. Would be nice, but we thought about this, but we decided not to go to this route because already the system is complicated. But maybe in the future.
If there's more question, you can ask Omer after the lecture.
Thanks.
Thank you very much.
Okay.